Good morning everyone. Today our group teen adventure would like to present about DNA, genes and chromosomes. These three components are very important in understanding how living things grow, function and pass straight from one generation to the next. Firstly, I would like to introduce members from Teen Adventure who will participate in this project. They are G9003, G9004, G9006, G9007, G9008 and G9018. In this presentation, we will explain what DNA is and how it works, how genes control traits, how chromosomes organize genetic information, and the differences between them. We hope this presentation will help you understand the basic structure of life more clearly. So let's begin. Definition of DNA: deoxyribonucleic acid is often called the master molecule of life. It is a long, helical biological polymer that contains the unique genetic code responsible for the development, function, growth, and reproduction of all known organisms. It holds the complete set of instructions or the blueprint required for an organism to build and maintain itself. And structure of DNA. The double helix model. The structure of DNA is characterized by the female double helix structure first described by Weston and Crick in 1953. This structure resembles a twisted ladder and is fundamentally built from smaller units called nucleotides. Nucleotide component. Each nucleotide consists of three parts: a deoxyribose sugar molecule, a 5-carbon sugar, and a phosphate group, one of four nitrogen-containing bases, and adenine, guanine, cytosine, and thymine. The backbone. The two side strands of the latter are known as the sugar-phosphate backbone, found by the alternating attachment of the sugar of one nucleotide to the phosphate group to the next. Base bearing. The range. The nitrogenous bases face inward, forming the range of the latter. They are held together by hydrogen bond according to the rule of complementary base pairing. Adenine always pair with thymine, guanine always pair with cytosine. Antiparallel strand. The two strands of the helix run in opposite direction relative to each other. One runs 5 feet to 3 feet and the other one 3 feet to 5 feet. This arrangement is essential for DNA replication and transcription. Function of DNA. DNA primary functions are central to life. Storage of genetic information. DNA stores the long-term hereditary information that detects the synthesis of all protein and RNA molecules required by the cell. Self-replication. DNA has the unique ability to make exact copy of itself through a process called DNA replication. This ensures that when a cell divides, each new daughter cell receives a complete and identical set of genetic instructions. Mutation and variation. Changes in the DNA sequence. Mutation introduce genetic variation, which is driving force behind evolution.
A gene is defined as a specific structure of DNA that contains the instructions necessary to produce a specific fractional product, most commonly a protein or a fractional molecule of RNA. Genes are the fundamental physical and functional unit of heredity. Definition of gene. A gene is defined as a specific structure of DNA that contains the instructions necessary to produce a specific fractional product, most commonly a protein or a fractional molecule of RNA. Genes are the fundamental physical and functional unit of heredity. 2.2 Structure and function of gene. Structure. A gene is located at a precise position called a locus or a chromosome. It includes the coding sequence that is translated into protein as well as non-coding sequence and regulatory region that control when and where the gene is expressed. Function. Gene expression. The main function of a gene is to determine the trait of an organism by controlling the synthesis of a protein which acts as an enzyme, structural component, or signaling molecules. This process, known as gene expression, involves two steps. Transcription. The DNA sequence of a gene is copied into a messenger RNA molecule. Translation. The mRNA molecules leave the nucleus and is read by ribosome, which use the code to assemble a chain of amino acids, forming a specific protein.
Definition of chromosome.
A chromosome is a highly condensed thread-like structure located within the nucleus of every eukaryotic cell. Cells with a true nucleus. It is essentially a single, organically long molecule of DNA tied around specialized protein cell histone. Structure and function of chromosome. Structure. The complex of DNA and histone protein is called chromatin. When a cell prepares to divide, this chromatin coins and supercoins further to form the compact, visible structure known as a chromosome duplicates itself, resulting in the characteristic H shape where the two identical hapless sister chromosomes are joined at the concentration point called the centromere. Function. Efficient packaging. Chromosome organize the immune lot of DNA into a small volume so that it can fit inside the tiny nucleus. The human genome contains over 3 billion base pair of DNA, which is condensed into 46 chromosomes. Accurate segregation. Using cell division, mitosis and meiosis chromosome ensure that the genetic material is accurately replicated and divided equally between the two daughter cells, providing genetic destroyer. Types of DNA, genes, and chromosomes. 1. Types of DNA coding DNA. Sequences of DNA that carry the instructions for making proteins. This makes up a superficially small percentage, about 1 to 2% of the human genome. Non-coding DNA. The vast majority of DNA that does not directly code off proteins. Much of this is involved in regulating genes expression, turning genes on or off. Some parts are still being studied of fashions. Type of genes. Structural genes. Code of proteins that form the structure of the cell or are involved in metabolism. For example, enzyme, transport, and proteins. Regulatory genes. Code of proteins that control the rate or manner of expressions of other genes, often acting as switches. Housekeeping genes. Genes that are essential for the basic maintenance of a cell and are expressed in almost all cell types. Types of chromosomes, autosomes. These are the non-sex chromosomes. Humans have 22 pairs of autosomes. They control genes for genital body characteristics. Allosomes, sex chromosomes. These determine the biological sex of the individual. Humans have one pair. XX is females and XY is males. The difference between DNA genes and chromosomes. The relationship between DNA genes and chromosomes is best understood as hierarchical organization, moving from the minute molecular instructions to the large structural package design for efficient transmission. Fundamentally, DNA disorderbodes access is the raw chemical material that allow helical molecule that constitutes the entry genetic code. Is the language of life, chromosomes or a specific sequence of nucleotide base that contain the complete blueprint for every component of a organism. DNA primary role is to store and replicate
this information faithfully. A gene is an entirely foundational composite, defined simply as a specific segment of the long DNA molecule. Then all DNA as a message ambiguous score or a text. A gene is a particular well-defined progress within that take the carry the complete instruction. Says for manufacturing one specific functional produce, usually a protein or a regulatory RNA molecule. While DNA carry all the information. The gene is the added coding unit that determines a single biological trait, such as eye, color, or the function of the specific enzyme. A typical chromosome can contain thousands of genes located linearly along the DNA stem. Finally, the chromosome represents the structural and organizational level, given that the DNA from a single human cell is nearly 2 meters long. The chromosome as a sophisticated packaging mechanism is a highly condensed visible cellular format when the long DNA strand is tightly coiled and spooled around splicated protein. Called histone, this robust composite structure is essential for two key functions: feeding the message genome into the tiny nucleus and, most critically, ensuring the accurate and equal segregation and transmission of the genetic material to daughter cells during cell division, mitosis, and meiosis. Therefore, DNA is the code, the gene is the specific instruction, and the chromosome is the organized package that delivers the instruction.
Configuration DNA genes and chromosomes represent a tightly integrated and fundamental biological system. The DNA molecule provides the informational raw material. The genes are the functional units that translate this information into biological action, and the chromosomes serve as the robot efficient organizational container or story and transporting the entire genetic library from one generation or cell and generation to the next. This created management of this heritage is critical or mainly driving evolution and assuring the stability of genetics inherited to unique product that or specific stories at the maturation or DNA propagation or definition of genetics discovered. Configuration DNA genes and chromosomes all work together to form the foundation of heritage. DNA stores the information genes carry specific instructions and chromosomes organize and produce that information. They determine how organisms load, grow, and function. Understanding this concept helps us learning more about biology, health, and how data is passed from parent to children. Thank you for listening to our presentation. If you have any questions, we would be happy to answer them.
Oh, wow!